Okay, here we go.
This is another very small unit. We're talking about an ecological niche. And a niche is basically a species' role in a community. So you could think of it like if a location or a habitat is the species' job site, the niche is a species' job. And so this includes things like the time of day or year that a species is active, what parts of the habitat does it use, what type of prey size does it take, so is it eating little tiny things or larger things, what temperature can it tolerate, and what trophic level it exists in. So the example I've got here is a ptarmigan, which is a small bird that lives in the Arctic. And the time of day that it's active is daylight, so this is a diurnal animal. The parts of the habitat that it uses are mostly on the ground. As you can see here, it's standing on the ground in the snow. The prey size that this bird takes are very small insects in the summer and possibly other things in the winter. The temperature that it tolerates is obviously very cold. It lives in the Arctic. And its trophic level is that of a primary or a secondary consumer. So look in your supplementary materials for more information and make sure that you're trying the practice questions involved with the niche and the habitat so that you understand what kinds of things you might be asked about them on the test.
I will. Not yet, but I will. Did it take? There's niche. Oh, there it is. It did take. Good.
This very short unit deals with habitat, which is pretty simple. It's a species' physical location, which is all of the biotic and abiotic factors that are involved in the place that it lives and where it needs to live to survive, the things that it needs to survive. So examples of habitats are ponds, forests, rivers, and grasslands, such as the one that you see this American bison grazing in. So check your text for more information and details about different habitats. In this unit, we're talking about community ecology. And the community ecology is the study of interactions among different species within the same area. And this includes topics like competition, predation, symbiosis, and succession. We're going to talk about each of these in turn. One example of competition or topics in community ecology that is of more and more importance as people change the surface of the planet is how do species like beavers, for instance, deal with destruction of their habitat, such as cutting down forests. Another important concept is competition. So this is when more than one species in a community attempts to use the same limited resource. So for instance, this ocotillo here is a type of cactus in the desert southwest of the United States, and it is competing for water with the other plants that are in the area. So we've got some creosote bushes, there's some other cacti here, and so competition is a very important topic in community ecology. The competitive exclusion principle says that no two species can occupy the same niche indefinitely because one will be a better competitor and the other will go extinct locally. So that means that basically two species can't try to do the exact same thing the exact same way at the exact same time forever because one of them will do it better than the other and the one that doesn't do it as well will go extinct.
And so
niche partitioning is an interesting phenomenon that allows species to interact with each other or to survive without competing with each other as intensively. And basically what this means is that the jobs in an ecosystem are divided and subdivided into tiny, tiny little bits so that one species of plant might be growing very tall and it might be using a lot of water, but water that's very deep into the soil with a taproot. Another species of cactus might be using a little bit less water. It has surface roots. And so those two types of plants are competing for water, but they're doing it in a way that they are allowing each other to coexist because the cactus has the surface roots, and so it's not competing with water in the same depth of the soil as the other one. So that's just one example of niche partitioning. Symbiosis is another important concept, and this is a close interaction between two species. Mutualism is a case in which both species benefit. Commensalism is when one species benefits but the other is not affected, or we say it's neutral. Parasitism is when one species benefits but the other is harmed. And I've got this picture here of termites because termites have a little protozoan organism, which is a single-celled eukaryotic thing that lives in their guts and actually helps them to digest the wood that they're eating. So the termites get food and the protozoan also gets food out of the deal. So this is an example of a mutual symbiosis because both are benefiting. And then we talk about succession. There are two different types. Succession is a process through which a community recovers from a disturbance. So for instance, this picture here is, you might be able to tell that these are volcanic rocks. And so these little plants that are growing here are some of the first that have colonized after the volcano erupted and covered this island in lava. So primary succession happens when there is no soil to begin with. So lava flows are good examples because after the lava dries, there's absolutely no soil and no living things there. And then a glacial moraine is a place where a glacier has receded and left a bunch of rocks. And so basically there's no soil and there are no living things there either. So primary succession happens in areas with no soil. And then secondary succession happens in areas like an abandoned field where soil is present and the plant community changes based on how long the community or the abandoned field has been left to itself to recover. So make sure you check the readings if you need more details about the differences between different types of symbiotic relationships and primary and secondary succession.
Okay. This time I'll do the supplemental reading first. 33, one page. Either 33 pages or one of 33. I don't know. I will look at it later.
In this unit, we're talking about population growth and regulation. And so, first of all, what does that mean? It's a study of organisms of one species that are living together, and we call this a population. So population ecology is primarily concerned with things that limit the size of a population, the birth rate, the death rate, and the growth rate. Biotic factors that limit a population are things that are alive, obviously. The biotic part meaning that we're talking about living things. So dispersal is one biotic factor that limits a population. The more individuals that leave, the smaller the population is going to be. Competition, predators, a lack of prey or food, and parasites are also living things that limit a population size. Abiotic factors are not living things that limit a population size, and these include things like climate, landscape, soil, the salinity or the saltiness of the water, and sunlight. Density is another important concept in population ecology, and this is the number of individuals living in one unit area. So for instance, how many penguins are there on a particular square mile of land? Density-dependent regulators are things that depend on the density of the population, and so they are more important for limiting the population when the density increases. So the more individuals there are, the more competition there's going to be, the more predation there could be, and the more likely there are to be diseases that are spread among the individuals. So density-independent regulators are things that do not have anything to do with how many individuals are in a unit of area. So things like climate, disturbance, and pollution are density-independent regulators. So if you need a little more information about the difference between biotic and abiotic or density-dependent and independent regulators, make sure you check the supplementary readings. And finally, we'll end up with demography and growth. Demography is a set of vital statistics, and that just means things that are relevant to things. So dispersal is one biotic factor that limits a population. The more individuals that leave, the smaller the population is going to be. Competition. Abiotic factors are not living things that limit density-dependent and independent regulators, making growth. Demography is a set of vital statistics, and that just means things that are relevant to the lives of the living things that you're looking at. So birth and death rate and age at maturity are important for that. Growth rate is how fast a population gets larger or smaller. So the rate of growth is R, and R equals births minus deaths, which we use the letter M for that because it's mortality. So rate of growth equals births minus deaths. And I have these two pictures here on purpose because orangutans and the Sumatran tigers are examples of animals that have very slow population growth rates. So put all that together and we've got basic principles of population growth and regulation.
Okay. Great. Did I do this already? I don't remember. Yeah, I did. Should count. Find out. Pretty pictures.
In this unit, we're talking about biogeochemical cycles, and that is kind of a confusing long word, so I thought we'd split it into parts so you can see what it means. So the bio part means that we're talking about living things. The geo part means we're also talking about sediments and rocks, and the chemical part means we're talking about molecules, and obviously cycles are circular movements. So if you put all that together, we've got the circular movement of molecules through an ecosystem's living and non-living things. So we're going to talk about 4 biogeochemical cycles. We're going to start with the water cycle. So there are several things that you need to pay attention to with this. You need to know where you can find water and where it goes is a red, and ponds are things that are circled in red surface of the earth through, and then it condenses around in the ecosystem and the underground water supply. The water cycle is and falls back down. Where you find and another is from the atmosphere and then carbon dioxide are one of the major sources of returning carbon dioxide to the atmosphere. And carbon is found in, so where we get the fossil fuels are in this fossil carbon. Major sources of it, the terrestrial photosynthesis return. Actually, the majority of gas in the atmosphere is nitrogen, about 80. So one way is that sediment and then other bacteria return nitrogen. So you need to know where to find nitrogen, such as denitrification and fertilizers. Terrestrial organisms take it out. Finally, we're going to finish up with the phosphorus cycle, and phosphorus is the only being represented up in the atmosphere here. You'll see that there is no phosphorus is the only one of these cycles that does not have a large component in the atmosphere. So you'll see that there is no phosphorus component being represented up in the atmosphere here. Of it is moving around in terrestrial food webs. Little of it moving around at any one time. It's moved around a little bit, but the phosphorus material for differences and similarities between these 4 different types of cycles.
We'll have to go over that for sure. Happens when I go back.
In this unit, we're talking about energy flow in ecosystems. And the term energy cycle also basically and found in an ecosystem. And so a trophic is removing food from the system. All organisms that do photosynthesis are producers. All organisms that do photosynthesis are producers. So this is an example of trophic levels of the trophic structure. These are the autotrophic organisms. Primary consumers eat primary producers. Secondary consumers eat primary consumers eat of the trophic structure. Example of trophic levels. Make sure you check the supplement producers. Consumers eat producers or other consumers. So this is an example of trophic levels producers. Consumers eat producers or other consumers. Consumers eat primary producers. Trophic categories are another way of thinking of these are the photosynthetic organisms from eating other organisms. Ecological roles include levels like Earth. The transfer of energy in ecosystems is actually very inefficient, and that's because organisms lose heat during the process percent of what the previous level had. So if we start percent of that, which is 100 joules, and then the secondary consumers of energy going up the trophic structure. So make sure you check your textbook or the book that came with the course if you need more information about that.
That's cool. I didn't know that. Only get 10% as they go up the levels. That is interesting stuff.
In this section, we're talking about principles of ecology. We've got geochemical cycle, habitat. We'll talk about energy flow, and then we'll get into biogeochemical stress. We'll do limiting biotic and abiotic. Talk about community structure, growth, and regulation, including competition. We'll also talk briefly about a niche. What does that mean? And some example introduction. And then we'll talk about dispersal and species diversity, immigration, and then age effects. Then we'll talk about evolutionary interactions, one-way interactions, and
this is Module 3.
In this 3rd unit, we're getting to my specialty, which is ecology and social biology, and with energy flow to biogeochemicals, population regulation, biogeography, and principles of evolution section. We'll talk about history of evolutionary concepts, concepts of natural selection, adaptive radiation, major features of plant homology and analogy, balanced polymorphisms, and evolutionary history of human typed and learned social behavior. And then we'll talk about human population growth in the natural world.
Oh, I'm not doing that test right now. Okay, did I do the section? Let's see. Green. Everything except for the check for understanding, which I'm going to wait on. Okay, and the introduction I did. Oh, I didn't finish these, but I did them. I'll do them again. I did one of those. I did none of those. Okay, let's go down low. Let's do these things real quick. So I think I just need to touch it, and it's fine. I listened to you before, and this before, so it should show up with the check marks after I go through this. Yeah, I saw these testimonials already. It Should let me scoot through, as long as I go through them. Okay, why am I way over here now? I should be in biology. It's not a perfect site, but it's pretty amazing, actually. Okay, now, where, oh, no, wait, oh, great, so next again. Does this have the outline? Okay, no. Ooh. Wait, did I do all this? It says I did them all. I don't think I did all of them. I see check marks up there. It doesn't seem right. Niche. Everything but the check for understanding. Okay. Population biology. There we go. Okay. I did not meet your professor. I didn't do any of that. Oh boy. Oh, there's a lot of more to go through. Oh yeah. Ooh. 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 Ah. Nope. Wait. Hey, I did those. I didn't do these. Ah. Do the summary.
Congratulations, you finished the CLEP biology course.
No, I haven't.
Now, with some review and practice, you'll be prepared to pass the CLEP exam and earn college credit for the class. We covered a lot of material in a short amount of time. We started off with molecules and bonds. We explored the cell as the basic unit of life and how it divides. We discussed genetics, including chromosomes and inheritance. Then we went through body systems of plants and animals, their reproduction and development, and finally into ecology, behavior, and evolution. Remember to review the text in the lectures, focusing on things you're having trouble remembering. To do the best possible on the CLEP test, try to master the information so it all makes sense. With that, thanks for taking the CLEP biology course with me, and good luck on the test.
Thank you. Not entirely true. I'm not quite here yet, but I'm allowed to skip around, so I'm going to skip around. I did not register. I'm not ready yet. I'm not ready for that, though. Oh boy. Oh gosh. Oh gosh. I just did 3, so let's do 2. I mean, yeah, I should do the check for understanding after each right away. I have to. No, I gotta do it my way. It's silly, but I have to.
In this short unit, we're talking about polygenic inheritance, which basically means that something is inherited, but it's not just one gene that it's inherited from. Polygenic inheritance means that there are 2 or more genes affecting the same phenotypic character. Human height and human eye and skin color are good examples of polygenic.
Yuck, peanut scores. Hello. Gracias. Thank you, thank you. Yeah, this portrait. I've been seeing so many of those skibbity toilet things with the camera head. I didn't realize I made camera head.
You just have an iPhone.
It's just literally an iPhone. You're right. It's just an iPhone. That's all it is. It's big swirly thing. 2 megapixel. It's very nice. 144p or 14, whatever. Is it 2 megapixel? I don't know. Whatever 1444p is.
The only camera that I have is that really old Logitech 720,
Yep, I know that one.
and it's like half broken.
It's
at this
potato,
point.
yeah.
It's so.
Yeah. Oh yeah. But it's like fine. I was like, it wasn't that much money, but I was looking at all the different ones, and I'm like, I want to have like sharp for a change. I'm tired of looking like a potato. And I tried the different levels, and the weirdest thing is, last night I did the when I hit the 1444 or whatever over 1080. There was an actual difference between 1080 and the other one. My face was pink. There was a color in my face that I've never seen on a camera, but it's sharp enough to catch it. It's weird. It does 60 frames per second at 1080p, but I like the sharper. I don't know.
That's cool.
It has a little privacy shutter for paranoid people. Apparently that's a big deal.
Oh, it is a big deal on the, especially on like laptops and stuff. You need to have that little cover.
Yeah.
Or else it won't sell.
I know. I guess it's I think, well, probably because big companies got caught spying, like actually turning it on when they weren't supposed to. Like maybe the software's gonna. Was that cat?
That was cat.
My God! I just see moving flap and nothing. Okay, but yeah, it's a big deal. They won't sell it without the flap thing. So
Yeah.
I don't know. But the rigging you guys are doing, I've been peeking. That rigging is incredible. The rigging, the guys like you do like the 3D shit, like the robots, the parts game. It's still going, isn't it? There's nobody watching, so it's probably alright. Yep, I'm still streaming. Drop 13 frames. Sorry. Hungry.
In this unit, we're talking about the chromosomal basis of inheritance. And so first, Mendel came up with two laws. One of them is the law of independent assortment that says that every possible combination of alleles is equally likely for each gamete. So in this picture, I've got circled all of the possible combinations of alleles that could come from this potential metaphase division. So we've got red with red alleles up here. We've got blue with blue alleles over here. We've got red and blue over here, and blue and red over there, which ends up being the same thing. The second of Mendel's laws is the law of segregation. This says that paired genes separate and randomly recombine into the gametes so that offspring have an equal likelihood of inheriting either one. So that brings us to concept of probability. If independent assortment is occurring, the laws of probability predict.
I got it. Oh, I hated this stuff.
In this unit, we're talking about Mendelian inheritance, and it's only fitting we start out talking about the man himself. Gregor Mendel was an Austrian monk who studied pea plants and their inheritance in about 1865. Inheritance is a characteristic that is passed from one generation to another in the form of genes. And Herr Mendel was the first to quantify genetic tests, which was really important because he helped us to understand how inheritance works. Mendel's ideas are still accurate because they are for simple systems. There are some non-Mendelian inheritance rules that Mendel didn't know about because the pea plants he studied were relatively simple in their genetics. He coined several terms that we're going to talk about. There are roughly 10 of them. We still use these terms today. So the first is an allele, which is an alternate form of a gene. Everyone has two alleles for each gene, and they're represented by letters. For instance, big R or little r. A homozygous individual has two copies of the same allele, so big R or little r little r. A heterozygous individual has one copy of each allele, so a big R and a little r. A dominant allele is always expressed when it's present, and it's shown by a capital letter.
Appearance.
So big R, big G, or big N. A recessive allele is always masked when a dominant allele is present, and we show recessive alleles with lowercase letters, little r, little g, little n. A genotype is the alleles carried by an individual, or its genetic makeup. The phenotype is the appearance of an individual, and a cross is sexual reproduction between different individuals. For instance, Mendel studied pea plants, and one of his most famous crosses was between peas with purple flowers and peas with white flowers. And finally, a character is a feature like an individual has, such as hair color in a person, obviously, or plant height in one of these pea plants. A trait is the genotype or the phenotype of an individual for a given character. So, for instance, a person could have red hair, or a plant could be either dwarf, being small, or tall, being much taller than that. So check your book if you need more information about understanding.
Yeah, I do. I will. Oh my god, I forgot all about that stuff.
This section is about principles of heredity, which is a fascinating topic. We'll start out with Mendelian inheritance and then get into the chromosomal basis of inheritance. We'll talk about linkage and polygenic inheritance. So let's look at each of these sections in a little more detail. With Mendelian inheritance, we'll talk about some terminology that chromosomal basis of genotypes, Mendelian genetics. These are things that he didn't know about during his time. So we'll talk pics, and then we'll talk about polygenic, multiple genes, and diseases.
Okay, you see, I did it. There's something wrong with how I did it, so. I'll be back. Ugh, here we go again. It's dreadful. Awful. Am I really doing it this way? I am. Okay, don't watch, just do it. This one didn't take.
The probability predict the genotypes of offspring from the crosses. So the parental generation, which we show with big P, is the parents of a cross between two individuals. The first filial generation, which we represent with F1, is the offspring of P. The second filial generation, which we show with F2, is the offspring from the cross between two F1 individuals. So, for instance, in this picture, our P generation is a cross between a pea plant with white flowers and a pea plant with purple flowers. We happen to know the genotypes. F2 is the offspring from the cross between two, and then when we cross two F1 individuals, you get the F2 generation, right, and your individuals can either be little p, big P, or big P, little p. And so we use something called a Punnett square to estimate the probability or the likelihood. Using this, big Y, little y, and then those with just two characteristics.
Okay, I will get back to it, I promise. This I did. This I did. Oh my gosh. Don't mind me. Oh.
Examples of polygenic inheritance. Diseases are also commonly caused by polygenic inheritance because polygenic traits are hard to predict. For instance, diabetes, heart disease, and hypertension inheritance. Read text for more information.
Okay, I will.
Human embryos develop their own blood vessels, so even though they exchange blood and other materials with the mother, the embryo itself develops its own blood vessels. So an embryo's blood vessels are next to the mother's, and the molecules are exchanged by diffusion. So since the mother and the embryo have blood vessels that are adjacent to each other, things like nutrients, water, oxygen, carbon dioxide, and other waste products can be diffused into and out of the embryo's blood. So for instance, the mother contributes water and oxygen and nutrients to the embryo's bloodstream, and then when the embryo needs to get rid of wastes like carbon dioxide and carbon dioxide and other wastes, those diffuse into the mother's blood vessels, and then the mother actually excretes those wastes. So take a look at the book that comes along with the course if you want to learn more information, or if you need some help understanding information.
This is actually interesting stuff.
In this short unit, we're going to talk about the formation and function of the mammalian placenta. This is formed from the outer cells of the embryo and the inner cells of the uterus, so it's like a cooperative venture between the mother and the developing fetus. It's the connection between the mother and the embryo. So in this picture here, the placenta is this pink surface that you see here, and the umbilical cord is connected to it and to the developing embryo. As far as the function of the placenta, it is for the transfer of nutrients and water and wastes between the mother and the embryo.
Great.
In this unit, we're going to talk about extraembryonic membranes, which pretty simply are the membranes that develop around an embryo during the process of its maturation. We'll start with the chorion, which is here and surrounding the developing embryo here. This regulates water, gases, nutrients, and wastes. In egg-laying animals, it's in contact with the inner shell surface, and in others, it's directly in contact with the uterine lining.
Lots to memorize.
The amnion, which is here, functions as a fluid-filled sac around the embryo for cushioning and for temperature regulation.
Coloring book for that.
The allantois, which is here, comes from the developing digestive tract, and it serves as a gas and nutrient exchange tissue. You also and all of the material.
Vocabulary. I'll go back to you.
In order to understand the development of vertebrate embryos, scientists have used several model organisms. So these are organisms that are easy to maintain in a lab setting and that have a standard developmental process that's really well understood. So we can figure out generalities about vertebrate development from these model organisms. Actually, the majority of what we know about the
Okay. Yeah, mice have lent us their toes. Now they have it.
So let's start this unit by talking about the zygote. This is a fertilized egg, and this is a stage from which all sexually reproducing multicellular organisms start their lives. What happens right after the zygote forms is called cleavage, when the zygote divides many times without changing size. So we start out here with it going from 1 cell to 2, 2 cells to 4, etc., until we end up with many cells. There are several developmental stages once the zygote starts to divide. The first one we call a morula, which is a solid ball of cells. The second is the blastula, which is a hollow sphere of cells. And then finally we get the gastrula, which goes through a process called and we end up with a gastrula. So remember, this in here is the digestive canal. Here, in addition to these short lectures that you're...
Oh, good. I know this one. More terminology.
In this unit, we'll talk about gamete formation and fertilization. Reproduction is complicated for multicellular organisms, and there are two processes involved. One is called gametogenesis, and the other is fertilization. So, in gametogenesis, we're making gametes, and basically that means sperm and eggs. In spermatogenesis, we're making sperm, and this occurs in the spermatogonia, which are cells in the testes. The primary spermatocytes, which are diploid, are formed, and then secondary spermatocytes, which are haploid, are formed from the primary via meiosis I. Sperm cells, which are also haploid, are then formed from secondary spermatocytes through the process of meiosis II. So here we can see the spermatogonium going through mitosis, and then meiosis I, and then the second station, the sperm and the egg.
Terminology.
In this section, we'll be talking about animal reproduction and development, and that includes gamete formation and fertilization, cleavage, gastrulation, germ layers, and organ system differentiation, experimental analysis of vertebrate development, extraembryonic membranes, formation function of the mammalian placenta, and blood circulation in the human embryo, and placental function.
Got it. Okay. Everything I need is in the slides, so it shouldn't be too hard, in theory. Oh, I'm not doing a test. Nope. Nope. I'm not just checking to see if I made it through. I did. I did the heredity. Yep. Doing it, not really. Boy, lots of terminology. Thankfully, I know some of it, but
in order to understand hormones in homeostasis and reproduction, we first need to understand the endocrine system. So this regulates the body's set points, such as temperature, heart rate, and metabolism. The triggers are important physiological events, such as puberty and reproduction. And the endocrine system facilitates cell-to-cell communication for things such as glucose uptake and antihistamine release, such as the stuffy nose and runny eyes and itchy face you experience when you encounter things you're allergic to, like dust mites or pollen. Hormones are chemicals secreted by an endocrine gland or an organ into the blood for the body's transport. So several examples of hormones include estradiol, testosterone, and cholesterol. Read the textbook that comes along with this prep course to learn more about the functions of each of these hormones. And done for breakfast or lunch or even dinner today, and join with a which release hormones depending on the animal. And they also trigger lactation or other.
Lots of terminology.
In this unit, we're talking about homeostasis, which is basically a term for a steady state. And this refers to the fact that some animals are able to maintain an internal balance. Some of them do it better than others. Examples of this internal balance are temperature, ion concentration, blood glucose, and blood oxygen. This elephant here, for instance, is a very large mammal. Amphibians. Check your supplemental readings for more information about the details of homeostasis and different types of thermoregulation.
Homeostasis is interesting.
All right, let's talk about major systems of the animal body. The animal body is a very complex machine. There are many things going on all at the same time, and all of these systems have to cooperate to maintain the living organism. I mean, think about all the different things that are going on in your body right now while you're watching this video and hopefully taking notes and paying attention all at the same time. Most of the body is controlled by hormones, but it's also affected by the environment. So the first system we'll talk about is the digestive system, and as you might have guessed, it digests things, processes ingested food and drink. The respiratory system is responsible for intake of essential gases and release of waste gases. Remember, you've got supplemental readings you can check on to find out more details about each of these body systems as we go through them. The circulatory food, such as bacteria, for when you go, again, check. In this unit, we're going to talk about structure and function in animals. We'll start with the major body systems, and then move into homeostatic mechanisms, and then talk about hormones and homeostasis and reproduction. So let's look at each of these sections: homeostatic components, feedback hormones, and how they affect homeostasis and reproduction, the endocrine glands in reproduction.
Nope, not doing a test. That one didn't take.
In this unit, we're talking about homeostasis, which is basically a term for a steady state, and this refers to the fact. So homeostasis relies, most fish and most invertebrates are all the details of homeostasis and different types of thermoregulation.
I didn't finish the video. That's fine. There we go. That took. This one didn't take yet.
In reproduction. In this unit,
Oh, words.
In this unit, we're talking about plant tropisms and photoperiodicity. A tropism is just a growth response that results in the plant growing towards or away from a particular stimulus. Phototropism is easy to remember because it involves light. So plants are responding to light. If we're talking about positive phototropism, plants are growing towards it no matter where they end up or how they're oriented when they end up there. Exposed to the majority of the wind don't grow very much, but the parts that are not exposed, such as vines, like you can see, zygmotropism. In a certain time of year, some of them will only or early in the autumn at the very end of summer. So check your reading materials to see if you need more information about specific tropisms and photoperiodicity.
I promise I will. I have to.
So we'll start out talking about plant growth and development by discussing some plant hormones in general and then specific types of hormones. Plant hormones have a similar function in plants as hormones do in animals, and they tend to be transported in the vascular system. Plant hormones control most aspects of the plant's growth and development, and there are these that you're seeing here in this picture, and they tend to be transported only from the tip to the base of a shoot. So you don't find these in the majority of the rest of the plant. Cytokinins stimulate cytokinesis. It makes it easy for you to remember what it is that their purpose is. They're produced in actively growing tissues such as roots, embryos, and fruits. Gibberellins affect cell division and elongation, fruit growth, seed germination. So as you can see in this picture here, slows the growth of plant tissue or injury, and such as fruit ripening.
Oh my God. It's ridiculous. Oh no.
In this unit, we're talking about plant gametes and fertilization. So, egg, the embryo is the growing zygote once it has gotten to the point of being more than a few cells. Plant sperm is formed by the male gametophyte, which you find inside the tissues of the pollen, which are all of these little yellow things here in this picture.
Yeah.
They're produced in very large numbers, and they leave the plant to join the eggs so that fertilization can happen. Transmittal of plant sperm depends on the species. Some plant sperm is swimming and travels from one wet area to another to find the egg. for more information on plant gametes and fertilization. Plants are unique among other organisms on the planet because they undergo what we call an alternation of generations, or you could think of it as an alternation of phases. The plant's life cycle is an alternation between haploid and diploid phases, and they go back and forth depending on their age, the season, the time of the day, the time of the year. Both phases can go through mitosis, but usually only one of them goes through meiosis. The sporophyte is the phase that tends to be dominant in most plants, but it depends on the species. So the diploid and haploid phases have different names. The sporophyte is the diploid phase, and the sporophyte, for example, pine trees, like the one you see here, the sporophyte is the one that produces spores, and it actually does that through the process of meiosis. And so the spores grow into gametophytes through the process of mitosis. And then the gametophytes then we call the sporophytes. And so you check the reading material about is producing spores, which come together to form a sporophyte again, which is back to 2. In section 2.2, we're talking about plant reproduction and development, and there are several units. We'll go through alternation of generations, gamete formation and fertilization, growth and development, and tropisms and photoperiodicity. In Unit 1, we'll talk about the alternation of generations, which is an interesting phenomenon that's unique to plants. So there are alternation of phases and
We didn't take? We seriously didn't take. Okay. Well.
to see if you need more information about specific tropisms and photoperiodicity. Ripening and programmed cell death. Gametes and fertilization.
Right. Everything but check for understanding.
This short unit is about food translocation and storage. So the food that we're talking about is sugars that were made through the process of photosynthesis. Phloem is the specific type of vessel that transports sugars from the leaves of the plant to the rest. So this diagram here shows you where sugars build up, which is inside the phloem here. Builds up in the phloem. For instance, potatoes, go back and check your supplementary transportation throughout the plant's organs. In this unit, we're talking about water and mineral acquisition and transport in plants. Before we get into that, the details of that, we need to understand that there are two major groups of plants, the non-vascular and the vascular. The non-vascular, etc. Vascular tissues plant through a process called attachment to other objects, and the tension are supplementary reading materials for more information.
I think I remember that.
For it. let's talk about plant organs. The plant's major organs include shoots, leaves, flowers, fruits, and roots. We'll talk about each of these in turn. Before we get to each of them, I wanted to show you a picture showing all of them together on a whole individual plant. So here are the roots, flowers, and fruits. Textbook, more about that if you're interested right now. Flowers and elementary material that came with this course. In this section, we're talking about plant structure and function. We'll go over plants, leaves, flowers, vascular plants, vegetation, and storage, where we'll talk about sugar movement. Talking about structure and periodicity. Cleavage, gastro of the mammalian of heredity, including Mendelian polygenic inheritance.
Yeah, there we go. Okay. Oh boy. What am I doing? Why am I doing it this way? I don't know.
Viruses are interesting because science has yet to determine whether they are alive or not. They have their own DNA or RNA. Some of them have one, some of them have the other. They can only reproduce in host cells, unlike living things, viruses themselves. Viruses are structured, they're inside this little sphere of protection to stop working yellow, and they are invading. Look for more details about that. Look for more details about that, and if you have any additional questions. Transformation is an interesting process. So remember that bacteria are prokaryotes. And this is one reason why they can become resistant, it can be changed. Transformation is when bacteria themselves have any of these red bits in it, but by the time transformation has finished, they've incorporated at least one piece of DNA from the difference between transformation and transduction. Transduction is when a virus, like this one here, which is specific to bacteria,
What's a pussycat? That's a sign to take a break.